Okay, today we are going to discuss about another genetic disorder, that is red-green color blindness. This red-green color blindness, it was first reported by John Dalton, John Dalton in 1798. For that reason, it is also known as Daltonism. Daltonism. Dalton was himself a sufferer of this red-green color blindness. So he first presented a paper on it in 1798. That is why we call this red-green color blindness as Daltonism. However, it was Wilson in 1911 who found the gene for red-green color blindness to be on X chromosome. So Wilson found it to be X-linker. Wilson found this disorder to be X-linker in 1911, right? So in our eyes there are there is a retina. In the retina, we have got two types of cells: rod cells and cone cells. Rod cells, they provide us nocturnal vision. They provide us nocturnal vision. While as cone cells, they provide us diurnal vision. So by the help of rod cells, we are able to perceive objects in dim light. And by the help of cone cells, we are able to perceive objects in broad daylight. So this night vision, it is known as nocturnal vision. It is provided to us by rod cells, and diurnal vision, broad daylight vision, color vision, that is provided to us by cone cells. These cone cells, as you know, they are of three types. Cone cells are of three types. One type of cone cell is known as chlorolabs. There is another type of cone cells. They are known as, the first type is known as erythrolabs. Erythrolabs. Second type is known as chlorolabs, and third type is known as cyanolabs. So there are three types of cone cells: erythrolabs, chlorolabs, and cyanolabs. Erythrolabs, they contain a pigment which is sensitive to red light. Erythrocytes, they contain a pigment which is sensitive to red light. So by the help of erythrolabs, we can perceive red light. Chlorolabs, they contain a pigment which is sensitive to green light. So by the help of chlorolabs, we can perceive green light. And cyanolabs, they contain a pigment which is sensitive to blue light. So by the help of cyanolabs, we can perceive blue light. So these cone cells, they are of three types: erythrolabs, chlorolabs, and cyanolabs. Erythrolabs, they contain a pigment which is sensitive to red light. So by the help of erythrolabs, we are able to perceive red light. Chlorolabs, they contain a pigment which is sensitive to green light. So by the help of chlorolabs, we can perceive green light. And cyanolabs, they have got a pigment which is sensitive to blue light. So by the help of cyanolabs, we can perceive blue light. So our color vision, it is because of the combined activity of these three types of cone cells. So we have got RGB type of color vision. So this RGB type color vision, it is by the combined effect of, by the combined activity of these three types of cone cells. Now, so we have got three types of photoreceptors. We have got three types of color receptors in our retina. So our vision is trichromatic. So we have got trichromatic vision. Since we have got three types of cone cells which perceive three different types of colors, so this vision is known as trichromatic vision. So we have got a trichromatic vision. But sometimes one of these cone cells do not function properly due to gene mutation. Sometimes the gene which is responsible for the formation of pigment in erythrolabs gets mutated. If the gene for the formation of pigment which is sensitive to red light gets mutated, that disorder is known as protanopia. That disorder is known as protanopia. Sometimes the gene which is responsible for the formation of pigment which is sensitive to green light, that gene gets mutated and the condition is known as deuteranopia. Deuteranopia. Or sometimes the gene which is responsible for the formation of pigment which is sensitive to blue light gets mutated and that condition is known as tritanopia. Tritanopia. So inability to perceive red light is known as protanopia. Inability to perceive red light is known as protanopia. Inability to perceive green light, that is known as deuteranopia. And inability to perceive blue light, that is known as tritanopia. In red-green color blindness, we are discussing red-green color blindness. Color blindness is of various types. We are discussing a specific type of color blindness, red-green color blindness, where the gene for perception of red pigment or green pigment that gets mutated. So in red-green color blindness, a person may be protanopic or deuteranopic. It depends upon which gene is affected. Mostly in red-green color blindness, the gene which produces pigment for perception of green light gets mutated. So we say red-green color blindness. It is a case of deuteranopia. So the person is unable to perceive the green light. Inability to perceive the green light, that is known as deuteranopia. When a person can only perceive two colors. So in red-green color blindness, when a person is suffering from a gene mutation, the gene which is responsible for the formation of pigment which is sensitive to green light gets mutated. Then the person has only two functional genes, or two functional cone cells, erythrolabs and cyanolabs. These colorolabs, they are non-functional. That condition is known as dichromacy. So when only two colors are visible, that condition is known as dichromacy. So we can say red-green color blindness. It is a case of dichromacy. Why dichromacy? Because the person is able to perceive only two colors in red-green color blindness. The person is able to perceive red and blue. The person is not able to perceive green, right? So when only these uh, two cone cells, these two types of cone cells are functional. So he's only able to perceive two colors. We call it as dichromacy. And when the gene for green pigment, gene responsible for the perception of green color is mutated, then that case is known as deuteranopia. So in red-green color blindness, we say this is a condition of dichromacy because the person is only able to perceive two colors, and it is deuteranopia because mostly the gene responsible for formation of this pigment which is sensitive to green light gets mutated. That is why we call it as a deuteranopia. So in short, we say red-green color blindness. It is a case of dichromacy and deuteranopia. Now, which colors are affected? A person suffering from red-green color blindness, he cannot perceive the colors correctly. So, which colors are mostly affected? A person who is suffering from red-green color blindness fails to differentiate between the color spectrum red, orange, yellow, brown, and green. A person who is suffering from red-green color blindness cannot correctly perceive these colors, ranging from red to green. So the person is not able to perceive red correctly. The person is not able to perceive orange, yellow, brown, or green color correctly. The person perceives these colors, observes these colors a bit differently. In this slide, a person who is suffering from red-green color blindness perceives red like this and green like this, right? And all the intermediate colors which are falling in between this red and green spectrum, they are not perceived correctly. So person who is suffering from red green color blindness perceives red like this and green like this, right? The normal colors are shown above, the red apple and the green apple. Below it are shown the colors which are perceived by a person suffering from red green color blindness. So this is the condition of a person who suffers from red green color blindness. So these are the problem colors in red green color blindness, right? Now we are concerned here with the inheritance pattern of this red green color blindness. The inheritance pattern of this red green color blindness is same as that in hemophilia. So a person who is suffering from a red green color blindness has the gene located on his X chromosome. So a female may be homozygous for red green color blindness. A female can be heterozygous for red green color blindness. Or a female can be homozygous recessive. Homozygous recessive. Homozygous dominant and homozygous recessive. So which is homozygous dominant condition? This XX, where both the alleles are normal. That condition is known as homozygous dominant condition. In heterozygous condition, a female may be having one normal gene, and on another X chromosome, she may be having this gene, mutated gene, which will be represented here by small c, right? In heterozygous condition, in carrier condition, the female has one normal copy of the gene on one X chromosome, and a female has got this mutated gene on her another X chromosome. Or the female may be homozygous recessive for color blindness, when she has got two mutated genes on both of her sex chromosomes, X chromosomes, right? So in this condition, where both the genes for red-green color blindness are mutated, then we call that female as homozygous recessive. If one gene is mutated, one gene is normal, then that condition is heterozygous condition. And if both the genes are in dominant state, both the genes are normal, then we call it as homozygous dominant condition. So a female can be homozygous recessive, color blind, or a female can be heterozygous condition, carrier for color blindness. What is the condition in males? A male has got only one sex chromosome X. On that, male may possess the normal gene. So this male will be normal. Sometimes on this X chromosome, male may possess the mutated gene. Then the male will be colorblind. So for color blindness, male is always hemizygous for the trait. For color blindness, male is always hemizygous for the trait. As I told you about hemophilia, the same condition applies for color blindness in males and in females. So like hemophilia, male is hemizygous for the trait, and like hemophilia, a female may be homozygous dominant, heterozygous condition, and homozygous recessive condition. So it is inheritance pattern is exactly same as that in hemophilia. Its inheritance pattern is exactly same as that in hemophilia. So let us make a cross. Let's make a cross. We take a normal female, we will cross it with a colorblind male. So this small c, it represents the mutated allele. So this is a colorblind male. This is a normal female. This is a normal female, right? So when the cross is made, we take the gametes. The gametes produced will be like this. Now we will fuse the gametes. The condition will be like this: X, 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 C. This X and Y, X and Y. We will cross now this X and this X. X, X, see this X and this Y, it will be X and Y. So we can see from this cross, this allele, it is inherited from males to her to his daughters. So this is a female, and this female is a carrier female. This is a son, it is a male, this male is normal. This is a female, it is a carrier female again. And this is a male, and this male is normal. So a colorblind man inherits his trait, inherits his character, the gene for colorblindness, into all his daughters. So all the daughters here, all daughters, here all daughters, 100% daughters are carriers. And all the sons, here in this example, 100% sons, they are not. So we observe that this gene for color blindness, it is inherited by a male to his daughters, and all his sons are separed. None of the sons inherits this trait from his father, right? Then how do sons get affected? We will see that in another cross. How do sons receive this trait? So we will see that in another cross. So in this cross, we observed that this color blindness, it was here present in the males. From the male, it went to the females, it went to her daughters, and all of his sons remained normal. So the father did not inherit this trait for color blindness, just this gene for color blindness into his sons. The father only inherited this gene for color blindness into his daughters, right? Now we will see in the second class how the sons are affected, how sons get color blindness. So now we will marry such a daughter, a carrier daughter, with a normal person. Now we will take a carrier female and we will marry him with a normal male. So this is a carrier female, this is a female, she is a carrier for color blindness. This is a male, this male is normal. So when they are married, the gametes are produced. Let us cross the gametes. The gametes fuse like this. So what do we see? This offspring, it is a female. This is a normal female. This offspring, it is a male. This is a normal male. This offspring, it is a female. She is a carrier female. And this offspring, it is a male, and it is colorblind male. So in this cross, it is clearly visible that males receive this trait from their mothers. Males receive this trait from their mothers. So this was a carrier female. She inherited this trait to her 50% daughters. To her 50% daughters, her daughters became the carrier. 50% of her daughters became carrier, and 50% daughters, they remained normal. 50% daughters remained normal. And similarly, this female inherited the trait to her 50% sons. 50% sons became colorblind, and 50% sons, they remained normal. 50% sons remained normal. So through these crosses, it is clear now that the trait is inherited from males to the females. The trait is inherited from father to daughter, and male does not inherit the trait to his sons. Then it is daughters who inherit this trait into their sons, right? So from females, the trait goes into males, right? So it indicates that This is mother, this is father. The trait was first present in the father, then father inherited his trait into his daughter, and sons remained normal. So from father, the trait went into daughters. When these daughters got married, they inherited this trait to their sons. And when these sons got married, they again inherited this trait to their daughters. Right? So in this way, we see that this trait, it moves from males to females, then from females to males. But in between two successive male generations, one generation remains free. One of the male generation remains free. So here it was present in father. From father it went into daughter. Sons remained free. Sons remained free. Sons did not receive the trait. Then when the daughters got married, they transferred it into sons. And they also transferred it into their 50% daughters. 50% daughters were affected, and here, 50% daughters were carriers and 50% sons were colorblind. Then from sons, it again went into daughters. And from daughters, it will again go into sons. So in this way, this trait moves from males to females, females to males, males to females, females to males. And in between two successive male generations, one generation of males remains free, right? So for that reason, we call this inheritance as skip generation inheritance. We call it as sex-linked inheritance because the gene for color blindness is present on sex chromosome X. We call it as crisscross inheritance because the trait moves in a crisscross manner. We call it as zigzag inheritance because the trait moves in a zigzag manner, right? So its inheritance pattern is exactly the same as in hemophilia. Now, are there colorblind females found? Yes, colorblind females are found. So how a colorblind female is likely to be formed? So we so far found that either the male may be colorblind or the female is wire carriers. Now we will see how a colorblind female is produced. As I told you, like hemophilia, it is most commonly found in the males. Hemophilia and colorblindness, red-green colorblindness. It is mostly the disease of males. Why it mostly affects the males? Because males are homozygous for the trait. Now let us cross a carrier female with a colorblind male. When we cross a carrier female with a colorblind male, we see the results. We see the results. These are the gametes. When we fuse the gametes, it will be XX. See, it will be X and Y. It will be XX, XX, C, C, and this will be XCY. So this individual, it is a female, but the individual is carrier female. This is a male, but the individual is normal. This is a colorblind. This is a female and she is colorblind, right? And this is a male who is colorblind. So colorblind females are possible. Colorblind females are found in the society, but their incidence is very less as compared to males, because colorblind females are not produced so much frequently as the colorblind males, because males are homozygous for the trait, and for being homozygous for the trait, males are produced in large numbers, and colorblind females are produced very less frequently. So the number of colorblind females in the population may be very less as compared to the colorblind males. So we say colorblindness, red-green colorblindness, it is again a disease of males, right? So like hemophilia, we have to remember some points here in this red-green colorblindness. The first point we have to remember in red-green colorblindness is that males are never carrier for red-green colorblindness. For red-green colorblindness. So like hemophilia, we have to keep it in mind that males are never carriers for red-green color blindness. Number second, this color blindness is more common in males as compared to females. As compared to females. Why? Because males are always hemizygous. Hemizygous for the trait. For the trait. Males are always hemizygous for red-green color blindness. Number third, a colorblind female. A colorblind female will always have a colorblind father, and she would always produce she was always produce colorblind son. So these are the 3 points which we need to keep it in mind for red-green color blindness. Males are never carriers. Males are never carriers. Because males are hemizygous, they have only got one allele. If the allele is mutated, they will be colorblind. If the allele is normal, then they will be normal. Color blindness is more common in males as compared to females. Because males are hemizygous for the trait, and females may be homozygous dominant, heterozygous or homozygous recessive for the trait. So color blindness is mostly caused in males because for being hemizygous for the trait. A colorblind female will always have a colorblind father. A colorblind female is always produced by a colorblind father, and she will always give rise to colorblind sons, right? So these are some important pointers which you have to keep it in mind for red-green color blindness and for hemophilia. These things are to be kept in mind. So I think our time is running out now. So this is session for today. Thank you for being with me.